Welcome to the session on regulatory engagement strategies for early stage developers. This topic is going to be on how we engage with the different agencies. We'll try to make it as global as possible. You know that the different agencies, the FDA, the EMA, PMDA, and all the others, they are very enthusiastic for early stage, especially for early stage developers, to get in touch with them as early as possible to map out your drug development pathway. And today we are going to draw on our collective experience in the field in cell and gene therapy throughout the years on how we engaged with the agency, what was the strategy that we needed to put in place so as to get more bang for your buck and get as much from the agency, their thinking, so that it could help the company or the sponsor to map out their pathway for their drug. With me, I have two excellent experts in the field. I have Lara Silverman from LIS BioConsulting. And Samar Mohanty from President and CSO for Nanoscope Therapeutics. We'll each introduce ourselves, and then we have a couple of questions we'll run through. I hope we can have some 5 to 10 minutes at the end for your questions. Lara, please.
Thank you so much. My name is Lara Silverman. Very happy to be here today. I'm actually a PhD scientist by training, and regulatory has developed over my career as my focus, as I realized that this is the gatekeeper to get us into the clinic, and it's where all the other work kind of comes together. So my background after getting my PhD was I ran an allo cell therapy company as the head scientist, set up a trial there in US and Japan, and for the past 5 years, I've been a fractional employee at a number of different early stage cell and gene therapy companies, and involved in quite a number of regulatory interactions. So happy to share my experience and perspective on the very early stage cell and gene therapy regulatory space.
Hello, everyone. Good afternoon. I'm Samar Mohanty. I'm also a scientist by training. Since we are developing therapy, which is not only mutation agnostic, gene agnostic, and disease agnostic, it required innovation in all forms, whether it's CMC, whether it's clinical endpoint. So even in regulatory aspect, defining those endpoints were very important. So I took over the head of regulatory also in our organization because I love innovation, whether it is science or in regulatory science. So we are developing this approach for multiple of diseases in the eye, retinal degenerative disease, and this will be the first mutation agnostic gene therapy that FDA will be reviewing. We have a rolling BLA submission ongoing there. We plan to complete in this half of the year, and we have got Sakigake designation in Japan and multiple designations in Europe. So we are, I think, going global with this program, and I'm looking forward to the conversation and share my experience on that.
So hello, I'm Shirley Bartido. I used to be in my past life the global regulatory lead for cell therapy at Takeda Pharmaceuticals. I come from 26 years of cell and gene therapy, starting with the early clinical CAR-T trials at Sloan Kettering, and going from auto to allogeneic CAR-T cell therapies with Selectus and gene editing, and then ending up at Takeda. I have at this point 12 INDs under my belt, shooting for a BLA sometime in the future, but would be very happy to discuss the experience I have witnessed during my 26 odd years in the field. So with that, I'd like to start posing the first question to our panelists. And we know there are a lot of mechanisms out there to approach the agencies for early interactions. And this, at least in the US, we have what we call the Interact. We have the pre-IND pathway. So the Interact is used if you don't have yet very much mapped out your preclinical studies and you would like to know what are the best approaches for your preclinical studies. The pre-IND is another pathway wherein you would submit a briefing book outlining your manufacturing, your initial preclinical studies that you have done, and your clinical plan, and ask the agency what are their reviews, what are their suggestions or comments on this type of review. And then you have with the EMA, you have what we call scientific advice committees, scientific advice meetings that you can schedule with them. It takes right now a lot of time juggling. So I here to try to get them, but I think the good thing right now is you can do a multi-country scientific advice and not just be for one country. So these are the different pathways, and of course with the PMDA, you can get also very early action. In fact, they want to have early interaction with the PMDA before you actually submit your CTN. So that was my experience, and that's why I would like to have my two panelists give their real life experiences in these early interactions with the agency.
I can start. I've done quite a number of interacts and pre-INDs in cell and gene therapy, and I actually spend a lot of time with my teams convincing them not to have those meetings. Normally you hear at these conferences, go early and often to the agency, but I think that's actually a mistake, because every time you meet with the FDA, it's a negotiation, right? You're trying to come to alignment on what they want to see and what work you have to do. And if you go to them with a blue sky idea and sort of let them lead the conversation, they're going to, of course, present the most conservative pathway, right? And we are very time constrained. We're very resource constrained, especially in this field with the funding environment. You do not want to be in a situation where they push you to do another animal model or another validation run. You want to go in there with a plan that's shorter and cheaper and faster, but with data to support that position. Because otherwise, they're just going to say, actually, we don't like that. You want to be able to say, you should agree with this because XYZ data shows us it's appropriate. So that's my first piece of advice when it comes to these early stage interactions. Actually, don't go too early. The second thing I would say is that pulling together things like interacts and pre-INDs, they're actually a really important business tool, because it forces every stakeholder in the company to sit around a table and contribute their part and reckon with the fact that, oh, the CMC team's not going to be ready to deliver material for this animal study, and we know FDA is going to complain about it. So I guess that's the reason I was drawn to regulatory, because it is a tool actually to help everyone get organized within the company and make sure that all of the moving parts go at the same time. So they're useful tools. They take a lot of work, a lot more work than you think they will. Not so much the writing of the document, but it's the sitting around the table and getting everyone on the same page. That takes a really, really long time. So you have to anticipate that when you sit down to do the work.
Yeah, I think I agree with Lara, you, and at the same time, you have to, before your process on the CMC side, you need to have certain alignment before you go in the path of having a process which you want to apply in clinical stage. But I want to move to the European side. So where I think scientific advice early on is important, whether you have to go to EMA or to a specific country is very important, like the country which you think has expertise on CMC side, and not like a pre-IND cross-functional meeting, but you can go for clinical advice versus CMC advice to specific country where you think there is expertise. For example, in cell gene therapy, Polyallelic Institute, or for clinical, you find the right expert who will give you guidance regarding multiple aspects. So unlike US, you have a pediatric investigation requirement when you go towards late stage, and that should not be getting. So you have to justify your mechanism of action or your drug target population and have some alignment there about use in the patient population. Otherwise, it can be getting, which we have seen in our program. Later on, if you go to the PEDCO and things like that, they will say, why didn't you come early on to us? That why you should be excluded from a pediatric plan. So I think it's important in different geography, the way they operate, so you need to interact with those agencies accordingly.
And just to give my 2 cents, fully agree with my panelists here, is that if you're a company or sponsor, biotech, or even academia, developing your first drug on a novel platform, I strongly suggest, if you have already enough data and everything, is to do definitely opt for a pre-IND. It takes a lot of work to prepare that briefing book, but it's well worth it because it really will set up the pathway for your next products developing on the same platform. And one of the strategies that I had applied when I was at Takeda, especially, was that in a pre-IND, you usually, the process is that you submit a briefing book. You first tell them you request it, then they'll say, okay, we'll grant the request. So you give them a little bit brief summary, make it exciting for them so that they will give you that option of having a pre-IND review with them. And then develop the brief. Then you submit your briefing book within within 60 days, and then they will usually come back with you with responses responses before your actual whether you requested to have a face-to-face or a teleconference or what we call they have an option right now, written response only. So depending on your confidence with the product, you can opt for any of these three. What I had opted, even for a novel one, was to do the written response pathway, requesting a written response only. So after the submission of the IND, after a certain time, I think within 3 weeks, they'll give you their preliminary responses. And when you have a written response, you are able to go over the preliminary responses and start asterisking or highlighting what are the different areas that you want to have a conversation with them to define what do they really mean by certain suggestions from them and everything, to get more clarity. So you pick those, and with the written response only, you actually get the option of a clarification period. And in that clarification period, so you send, okay, this, we agree with this, but this is the areas that we want clarification on, or this is the areas where we don't agree with you because of this, and this. Maybe they might suggest an animal model, which is not in your experience. Remember, we are the experts more than they. The agency learns from us more than we learn from them. And the more that we can tell them and justify our positions, the better. The clarification period comes after 21 days, and then they will tell you again, they will give you another piece of paper with all the responses. But this strategy allows actually for 2 complete reviews by the agency. Unlike a teleconference where you have 1 complete review and then you have to prepare and stress yourselves for a 1-hour TC. This way you're relaxed. You have time to ask the clarification questions. They have time to review, albeit it takes 3 weeks, but I think it's 3 weeks worth. So that is my experience. And with PMDA, since we are a Japanese company, it is better they have different committees for QA, different committees for CMC, a different committee for clinical. It is recommended by them to approach these committees early on before applying for the CTN.
Can I just touch on one thing there?
Yeah.
So this topic of meeting versus written response, even if you are planning to have a meeting, typically most of the things you ask or the information FDA gives you, the written response actually will be sufficient. If you write a good briefing package, they know what to respond and they know what they need to say to help you. I think the time where the meetings are actually helpful is exactly what you touched on, which is when we are the expert, honestly, and we need to help them understand something. So last year I had an FDA meeting and they didn't understand this very complicated animal model. Maybe we didn't explain it well enough, or maybe they're just really busy, I think, actually. And so in the meeting, we were nervous. As you said, it's an hour. Everyone's very stressed. It's not a pleasant experience still to this day. I get so nervous. But we were able to put the non-clinical expert on the call and have a peer-to-peer conversation with that reviewer and explain why the original plan made sense and what FDA proposed didn't. And it was very scientific. And at the end, the reviewer was like, oh, now I understand. Yeah, why don't you proceed with your original plan? So it very much is a collaborative conversation. They want to hear what we have to say. They want us to do our plan. They're rooting for us. And that's where those face-to-face meetings can be helpful. If it's just you're clarifying something or it's a nuance, I would leave it. Don't push it. They're bureaucrats. They're wonderful people. They want to look good too, right? You don't want to make them look bad in these meetings. So there's a lot of considerations with the interactions that you have with them.
So now that we have had 20 years already of 20 or plus years of cell and gene therapy. So there's been a lot of learn between sponsors and the FDA, both have, and the agency, sorry to say, that have learned a lot from in this field already. So my next question to, again, the panelists, our experts, is that do you have to go to the agency? What type of products now do we need to have this time-consuming, a lot of resources to put together to go to the agency and have, let's say, a pre-IND or to have these initial engagements? Has the landscape changed so that we can be comfortable with just going straight to an IND and not necessarily going through an initial interaction?
So I always think you should do a pre-IND. Never do an IND without a pre-IND. I'm with you on that. But I don't think everyone needs an interact. We have a tremendous number of guidance documents out. We have a lot of precedents. You don't want to walk into an interact and ask questions and they write you back and say, oh, just look at this guidance document. That's like, if as a regulatory consultant, that's a bad day at the office, right? So definitely do a pre-IND, less about the interact. And I think part of it is also setting the expectation of what you want this program to be. Is it an ultra-rare disease and you're going to be doing a trial with 5 people and these kids are going to die? That's very, very different than you're treating osteoarthritis and you're going to be treating hundreds of thousands of people. And the truth is FDA is going to give you different feedback based on what the target population is and who you're intending to treat, what the size is going to be. So the more information you can provide them, the more fruitful, in my experience, that conversation can be. And it also will vary if you're looking to do an IIS, very hot topic right now to do an investigator-initiated sponsor. They're cheaper, they're shorter, they're faster. And the interactions you have with the agency will be materially different if that's the path you take.
So, Samar, you have late-stage development expertise. Regulatory interactions do not just start at the very beginning before an IND. We do have interactions after IND. Can you allude to some of those interactions that you have had with the agency?
Yeah, definitely. So I think you talked about written response, but when you are in a late stage program, I think that time we are most fruitful in the personalized interaction where leadership is also attending those meetings in the director level and stuff like that at OTP. So that has been very helpful because there we also bring the patient voice to the decision making. Whenever it's about an endpoint, novel endpoint, you have to understand that this is completely new. FDA has never evaluated product, for example, in our case, very low vision patients. So taking the patient, having the patient voice was very important. They understand just maintaining their vision at that level is very important. Forget about improvement. So that's very important. And so I think you asked questions about pre-IND. We had to discuss about the clinical meaningfulness. Even we did a trial ex-US, and we bring that like investigator-initiated trial and bring the data here. So it was very important to get aligned on our phase 2 trial, whether the meaningfulness of the clinical endpoint and things like that. So it was very important that we show them what it means. It is a novel endpoint. So I think pre-IND definitely you have to do no matter what when you are first time doing a trial in the US. It becomes the complexity of the meeting becomes, it depends whether it's like after IND we have very lot of focused meeting on clinical and statistics. CMC meetings, they provide a lot of guidance. Our endpoint for potency assay, it was very, very important to discuss with FDA, make them understand it's not just a standard gene therapy. You have to put light onto the object and see the changes in the cell. It's a very complex potency assay, which to explain to them how it works also. We could not do it in a written manner because we had to explain. They were saying, oh, how do you put light on this flow cytometer on this? And it was too complex. Like 5 stages of processing the sample, 3 different chemical, you have to add at 3 different time points. There's a lot of variability. So I think in-person meeting helped us a lot in the late stage of interactions to make them understand how the potency assay works, how this novel endpoint works, and things of that nature. So I think They also saw the emotional component of the high unmet need of the patient. So all those contributed to get an alignment with FDA with some concessions in different respects.
And just to make the audience aware, you might be already aware. So after the IND, the other engagements that you have available to react with the agency is your end of phase 1 meeting, end of phase 2 meeting. In the end of phase 1 meeting, you would discuss the clinical, most likely your clinical data that you have accumulated based on the safety and efficacy of the product. And then for end of phase 2, usually if there needs be, this is where you will discuss your pediatric plan, what is your plans for maybe most like an accelerated approval. These days, the FDA, especially FDA, is not anymore satisfied with just, you take an accelerated approval and the company can run with it. They will now require that at the time of an accelerated approval application, you should be running your RCT, your randomized clinical trials with a standard of care as your control arm. So there are many different things that are being discussed at every stage of engagement with the agency. Usually it is the preclinical and manufacturing at pre-INDs. At end of phase 1 it will be your clinical. At phase 2 will be now your future plans for your pivotal or registrational studies, as well as any designation you would want to get, a breakthrough, an RMAT, and that's what you would be discussing. So be aware of what are the areas of discussion at every level of engagement. Very good. So, which is kind of I would like to get into a topic that seems to be the buzz these days, at least here at this area, and that is the new trend of cell and gene therapy going to an in vivo cell therapy process or procedure, as opposed to the ex vivo cell therapies that we have been used to in the last 20 years. What do we see as areas of engagement with the agency, especially if they're dealing with, we would like to propose a novel in vivo cell therapy panel?
All of these products at the end of the day have to meet the 210-211s, 1271s. I mean, they're all meeting the same regulations. If it's a gene therapy, a cell therapy, a tissue engineered product, it's a combination product, you have to pull in the 820s. I mean, at the end of the day, we're following the codes and then we're meeting the regulations. So if it's ex vivo modified, if it's in vivo gene therapy, a lot of the principles are still the same. Agency wants to know the product is safe, it can be made safe, and they want to understand the safety profile when it goes into humans. So I think that the regulatory process is not so different. Certainly the CMC effort is much simpler, right? But ultimately from the face of the regulator, we will downplay it. We will say this is just another thing, right? And it meets the regulations, no big deal, right? We don't want to sort of flag it as something big and scary. And in my experience so far with these more innovative or novel approaches, that's where Interact certainly is very useful, particularly to vet out the non-clinical program, because that's where you could really add a lot of timeline to your program. If you suddenly find out you have to do non-human primates, you need to raise an extra $3 million and add a year to your timeline, that will sink a company, right? You need to know that really, really early on. So that's where Interacts are helpful to lay out that strategy. I think CMC's come a long way. There's not a lot of surprises there anymore unless you're really doing a novel tissue engineering approach or delivery approach. It's really the non-clinical that adds risk for novel modalities.
So I'll add to that, there's a scientific thought that I think in vivo, when you are doing, it's very important. Let's say ex vivo, you are modifying the cell with a particular vector, or whether it's vector or with a non-viral vector.
The distribution in vivo will be very important, so I think that time the requirement will be much more, whether it's if you're doing any genes, if it's a vector, is there any integration with a different tissue, non-targeted tissue, so that's becoming very important. I think that's a big factor that we have to take into account in the non-clinical experimentation that we have to show.
Yeah, so definitely in this field, and I just came from a discussion actually this lunchtime, is that if you were to go into the in vivo space, I think interact will be, I mean, the best way is that I think the best of all because of its very novelty. It's you need to address the translatability of your animal models, as Lara had mentioned, because that's gonna affect your pockets, right? How much are you gonna need to carry on your studies? But not only is it true for in vivo, but also there's a lot of other things that are you can approach the agency as well, because right now they have allowed for a platform designation process. If you are planning to build on a common platform for all your drug development in the future, it will be worthwhile to already discuss at that early on how you would address the platform. Can you go for a platform designation so that in later your linear drug development, you don't have to go through the whole area of the same preclinical studies and everything. You would use that platform designation and be more focused on the questions you would ask the agency. And of course, asking the agency on your clinical plans, your safety monitoring plans, is that sufficient for every different phase of your drug development process? Make sure, to me, when you approach the agency, any agency, I prefer to be truthful as the regular. That is what I recommend my team, to be truthful and to be transparent. It's not going to serve your while if you want to, I wouldn't say hide or not disclose certain things that I think would be very important. You have to understand the agency is your friend. It is not your enemy. They are much entrenched and into this field as you are, and they want to see it succeed. And like I said, they learn from us and we learn from them. So it's a two-way process, and that can only be achieved by transparency from both sides.
Okay, I have to build on that because I feel strongly about it. There are two camps of regulatory strategy. One is to only give the information they absolutely need at that moment to answer your question. And the other is to be a bit more of an open book and be a little bit more vulnerable, I would say, to them. And I'm with Shirley. I advise my groups to be open because they want to help us. They want to see these products make it to patients. They're there because they care. It's not a glamorous job that they have, right? So if you tell them early on, look, our goal is to run one clinical trial, they know they're going to need to help you run that one trial, and they're going to look at your validation plan, and they're going to look at stuff that they wouldn't look at if it was an IIT. So there are different strategies. All of them are appropriate and right. So I'm not knocking anyone that has the other strategy, especially in some other countries. I've heard you might want to take a different tact, but certainly here in the US, my personal experience, early stage cell and gene therapy, it's never bitten me to be open and transparent. I know so far.
Yeah, I agree 100%, but I think we are not covering CMC as much. So what I'll say is it's very important that you have clear dialogue with them, that what's the scale, your commercial scale going to be, and how we are going to have a comparability. So you had when you're going initially for IND, you are having at a scale, but you should have a path forward to scale to what will be ultimately the. So for our case, it's a very low injection volume. So more preferred path is to run batch, more number of small batches. So you need to have experience on small batches because that will help you when you go late stage. And the second important part is your strategy for comparability has to be at the beginning decided, because if you don't have enough returns, you cannot do comparability. Same thing about potency, like endpoint, potency, and your titer determining assay or whatever assay which determine the critical quality attributes. You need to have alignment with the FDA, because nowadays a lot of time adaptive clinical trials with phase 1, 2, you can make it, if it has a now, of course, we are hearing a lot of CRLs because of lack of the conclusion. A lot of gene therapy had been going from phase 1, 2 to 3, and then that single trial can be a pivotal trial and give you approvability if you have the right alignment with the agency. And that includes also another point about natural history of the patients. So you have to collect in the PIT, you have some diseases you need to have the natural history study. And you cannot just go to, after your trial, you say, wow, like we the patient improved, their digestion improved, no headache, or this and that. But you have to have those pre-specified endpoint. They may not be primary endpoint, but you have to have pre-specified endpoint measured at multiple time points in baseline or screening, whatever, defined. It may be 20 different endpoints, but you need to have those evaluated in a structured manner. So that when you go to agency after your trial saying that, hey, look, in one or two of these endpoints, it improved. I know it's not always practicable to do all the endpoints, but you cannot just, what I'm hearing from agency interaction, you cannot go and let's say, oh, the patient improved with this, but I did not measure it prior to this, but he was having difficulty, he or she was having this difficulty, that difficulty. So agency wants something, if you want to apply the Bayesian statistics, now agency has said gene therapy has been very involved, released 3 different guidance documents, innovative clinical trial design, how to do post-approval monitoring, post-approval clinical data, and on CMC side also agencies showing that 3 deaths per PPQ may not be necessary. If you have a number of small batches in the PD and other things, you can show the manufacturing history. Agencies showing flexibility, but you have to do it in a scientific way. Whether it's about your endpoints, may not be primary endpoints, whether about your process development or about clinical endpoints.
The agencies expect for you to do your homework.
Do not expect them to do that homework for you. They will only comment on what you give them and nothing more and nothing else. So if you are, the more you give them, the more they will comment on you. And another thing that I want to say is that do not be afraid to push back. If you remember, you are the experts. You know more than they do. If you have a very sound, I tell my team, they tell me, can we push back on this? I said, okay, give me your rationale. Give me your justification. If I can buy it, then maybe they can buy it. But if I don't buy it, you'll have to go back to the drawing board. But do not be afraid to push back. I would one of the lessons I would like to put, and because I'm talking about that, and these days it's not enough just for the company or sponsor to think of your phase 1 trial. You may think that this is all encompassing, it's enough to keep you awake at night and everything, because at pre-IND you will, the agency will want to have an idea, what are you going to do beyond your phase 1? So in my experience, it's a good idea to give them a kind of a roadmap. Like, this is what we're, after the phase 1, are we going to do a phase 1, phase 1B? Are we going to do a phase 1, 2A? What is what population, what number of patients? When are we going to go global, etc.? It will give them an idea that we are really thinking about the drug development pathway of our product. And sometimes they will comment, sometimes we will ask a question, oh, can we apply for an RMAT after phase 1? They'll come back and say to you, yes, sure you can do, but we want to see the data. But if they tell we want to do this at phase 2, we want to do this at end of phase 1, it will give them a good idea, because they will tell you actually, okay, before a BLA, you need to have a validated potency assay. You need to have for your viral vector, you'll need it. They already spell it out at the very beginning. So if they can spell out already what future expectations they want from you as a sponsor, you can do the same. I don't know if my other panelists share the same experience.
Yeah, I think that's really critical. And what I've seen now, having worked with a lot of different groups, is there really isn't a right way to approach a BLA. I've seen groups that literally put a postdoc in a clean room and culture their cells in flasks. And then I've seen groups that spend $10 million to put everything in a bioreactor for their phase 1. There's lots of different ways to get to the finish line here, but it all is interwoven, especially when it comes to the regulatory pathway in the clinical trial program. And I think that's especially challenging right now, given that we're getting a lot of mixed signals from the agency on what is required for a BLA, particularly for the clinical trials. And the thing with these clinical trials is it's it's not an animal study. It's years of planning and tens and $20 million that they're investing, and you can't go back and redo it. And so that's a real challenge that we face as an industry right now until we're clear what the agency wants to see. And when they commit to something, they actually stick to it, which they're not. That the BLA stage is where it's really challenging. In the early stage, honestly, the stakes are a bit lower. You have to redo an animal study. Okay. You have to add an engineering run. Not such a tragedy. I think where it's becoming to be really tricky is in the later stages. And the more information you can give the FDA, the more you can have those conversations, and they can give you the right advice. They're choosing what to tell you. Like an IIS, they're not going to think about later stage manufacturing. They're just going to address safety of that product. So I think it's really critical to have that open dialogue with the agency.
Yeah, you talked about manufacturing. They expect in cell and gene therapy, they have all expectation that your manufacturing is going to change. It's going to develop. But the sooner you can get through that evolution of manufacturing changes, hopefully at the end of phase 1, the better it is for the less comparability studies they will ask you to do. If you keep on changing your manufacturing from phase 1 to phase 2, and then in BLA, there's another manufacturing change, they are going to hit upon it. So the sooner you can lock down on your manufacturing, at least for cell and gene therapies. And that's why early stage development, I think, is critical for cell and gene therapies, because this is where you will lock down. If you can get manufacturing out of the way, like Lara says, then they will just have to pay attention more to the safety, efficacy of the clinical data, and you will be ready for your BLA and have them coming to you and say, "Oh, you changed this, you changed that. Now we want comparability assays studies now across the board." That's gonna push you back big time.
Yeah, I agree. If you have two trials, one doing one process, like happened in Sarepta, you have seen 2-column chromatography versus other. So, and you change the site, manufacturing site. So the FDA has to interpret the clinical data that why there is a difference, variability. Already in this, there is variability. On top of that, you introduce new variability about manufacturing changes. So that becomes very important. I think I have seen, like we have done a lot of mistakes, like endpoints. Like we went with our phase 1 to a trial with endpoints not validated, let's say, and we think, oh, this is very cumbersome on us. But I have seen now I'm helping our own endpoint, which we refined, and we gave it to other cell gene therapy manufacturers in Europe, and then they did a very good validation study in the patients. And I'm jealous because they learn from us, and we help them, and they are doing the validation before they start their phase 1. And we went to phase 2 randomized trial without validating the endpoint. I'm just telling that a lot of mistake we do. We think, okay, we can move faster without doing this validation of your CMC assays. We don't have to do all validation, but potency, qPCR, titer. So those are very important. Same thing, your clinical endpoint. Even if it's a novel endpoint, you have to collect some data and take the time to collect data and then move forward. Otherwise, it becomes very challenging. I think we learned from our mistake, but yeah, we are able to navigate. But I'm seeing the other developers are using a very good path.
So before we open the questions, I'd just like to say one thing. Don't think that what the agency asked from previous sponsors way back when is going to be the same for you. There's a lot that has transpired. They know a lot. Remember, the agencies know all the clinical trials. They know what each company is doing. They know their pitfalls. They know what adverse events they're seeing. So they will maybe come back to you and say, "Oh, did you ever consider doing determination of like one time HHV6, HHV7, HHV8?" And that was not in the panel for the viral clearance. And we said, why are they asking about HHV, which is very prevalent in the 95% of the population? We found out later on, it's because they saw adverse events in another trial related to HHV. And there's a lot. Keep on top of the guidances, because now they expect more out of your clinical trial protocol. They're expecting now patient-reported outcomes to be starting to be gathered, even in early phase 1. It may not be comprehensive, but they want you to start. They want you to start on potency, for example. So they want to make you start earlier. So be in touch with your guidances. The biomarkers, they may be qualified, but they would like show them your evolution. They would like to see biomarkers now and everything, minimal residual disease and everything. All your qualified assays. Any other final words from our panelists?
I might just touch real quick on the concept of global trials. I know you have a lot of experience with it. I would say don't underestimate the effort of going to another country. Everyone's running to China and different places because it's less expensive, but there's so much challenge associated with language barrier, time change, cultural changes, that at the end, maybe it's cheaper, but it's really, really distracting and challenging. The other thing is that the regulators, they're trying to harmonize, but they're not harmonized yet. And so it's tricky if you get into a situation where one country demands one thing and another country demands another. You do need to kind of think that through and not get into a situation where you have two different processes or two different development plans that you have to put together. So yes, be global. It's the way to go, but it's tricky.
They want to do that, and I think the FDA and EMA want to do a Project Orbis. They haven't started it, but they want to do a similar Project Orbis among the cell and gene therapy to start getting harmonized. I'll see it when I'll believe it when I see it. Samar, any last words?
No, I think we discussed many topics. We should give the audience to ask questions.
Okay.
That will be
Okay, audience participation time. Any questions?
Hello. There we go. Thank you so much, panelists, for discussion. So my name is Adam Leszczyk from Blue Rock Therapeutics. We have a couple of products through early and late stage. Lara, to your comment about the going global. So we've actually had some success in Australia, right? So that was like one market that from a regulatory perspective we had some good penetration. Maybe a basic question here, but what in your experience, maybe Lara, this is more to you, in your experience, could you share maybe an anecdotal example or two on something that's commonly overlooked and that you've seen the same mistake kind of over and over, and it's common, it seems obvious, but it's very overlooked?
Okay, yes, absolutely. So my head was thinking about a global challenge that I've seen. When you go to Japan, the ancillary material requirements are different than in the US, and they're actually much higher. And so if you build your CMC process in the US for the US and you go to Japan, you're going to be in trouble. So that's one thing I've experienced before. That's really no fun. I would say in terms of other patterns that I see, one is the one I touched on before, going too early and letting the FDA push you into doing something you don't want to do and taking more time and money than you want to. I spend a lot of time talking people out of regulatory filings, which is weird because that's the work I do. I think that's the biggest mistake I see, honestly. And you need to do your homework, to your point. You need to read the guidance documents, see what's been done in the past. The field is quite mature. We have a lot of examples out there. And yes, your drug product is unique and special and amazing. But really, there's probably 10 other products that are almost exactly the same. And the more you can make them look like that, I think the better. There's always unique things, and that's what still makes it fun. But there's a lot of patterns you can follow. I would say the other thing is a common mistake or challenge I see is not being disciplined by using quality by design. Quality by design language is what the agency expects you to use. And by the time you're in phase 2 and phase 3, that is the language you're going to use. That's the process that you will be using. You need to be diligent and start it early, because by defining your CPPs and CQAs, it gives you a tool in development to move as quickly as possible to your final defined process. And again, it's what the agency expects. Your IND, your pre-IND is going to have that language in it. So I think that's another common flaw I see to implement early on.
I just want to add about global studies. I can understand why it's needed. You need to be For proof of concept studies, you want to not spend too much, and to do that in the States right now is very expensive. So you're looking at other countries, but please be aware. Remember, your goal eventually, the U.S. market, which is still the biggest market. So know that if you do your studies outside, there is a requirement at least by the FDA. You need to have a certain representative population. Now it's 25%. They want it actually more. So most likely you may do a phase 1A study for proof of concept, but think already that you will probably be doing a phase 1B or required to do a phase 1B in the United States before you can go to your global phase 2 trials. I think that's all we have for questions. We're right down to the last 10 seconds. We are around if you have any other questions, but thank you very much for being our audience today. Have a good rest of the week.